Do we have audio? Audio? Yes? No? Maybe? Okay. Are you ready? Do I have a thumbs up? I now call to order the Society's 2,390th meeting in the 148th year since its founding in 1871. Good evening, everyone.
Good evening.
My name is Larry Milstein. I am the president of PSW Science, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members and guests to tonight's lecture by Andrew Knoll in the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C., including our members and friends around the world who are watching the live stream of tonight's meeting on PSW Science's YouTube channel. We'll begin with a few announcements, a reading of the minutes of the 2,389th meeting, and Thomas Zurbuchen's lecture on eclipses. And we will then turn to this evening's lecture, followed by a question and answer period. Thereafter, I will present a small gift to thank our speaker, make a few closing announcements, and adjourn the meeting to the social hour. Please join me in thanking, well, first glitch of the night. Not sure why. Oh my. Okay, well. Okay, well, we may have to be manual tonight. So please join me in manually thanking the sponsors of the Fall 2017 and Spring 2018 lecture series, the Policy Studies Organization in cooperation with the American Public University, and a generous sponsor who has asked to remain anonymous. Well, what happened to the rest of the slide? This is really awful. There, that's what it's supposed to be. I am pleased to announce that the following new members have been elected to the Society. Collier Crouch, a Navy test pilot who is an instructor for FA-18s, interested in aeronautical engineering and testing, space exploration and organizational design, who comes to PSW through a family connection. And tonight's speaker, Andrew Knoll, whose interest will be apparent from tonight's lecture, in part. Please join me in welcoming them to the Society. Any of our new members who are here tonight, who have not done so already, are invited to pick up your signed reprint of Volume 1 of the PSW Bulletin from 1871 for the next few years. To get your copy, please see me after the lecture. James Heelan, our recording secretary, will now read the minutes of Thomas Zurbuchen's lecture at the 2,389th meeting 2 weeks ago.
March 9th, 2018. President Larry Milstein called the 2,389th meeting of the Society to order at 8:03 p.m. in the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C. He announced the order of business, that the evening's lecture would be live streamed on the internet, and welcomed new members to the Society. Recording Secretary James Heelan read the minutes of the previous meeting and the lecture by Christopher Ralston, which were read and approved without correction. President Milstein then introduced the speaker for the evening, Thomas Zurbuchen, Associate Administrator of the Science Mission Directorate at the National Aeronautics and Space Administration in Washington, D.C. His lecture was titled, Eclipses, Using Shadows to Shed New Light. Zurbuchen began his lecture by noting that science teaches us more about ourselves and about our world that profoundly affects us, sometimes in surprising ways. To illuminate his point, Zurbuchen presented 3 stories in which scientists learned about the world through eclipses and the absence of light. The first story was about the great eclipse of 2017. Zurbuchen said that our ability to predict space weather, which in turn affects terrestrial weather, is dependent upon our understanding of the sun. Many perturbations affecting space weather occur in the sun's corona, the solar atmosphere, where it is much hotter than the sun's surface as a result of magnetic fields. Scientists can calculate those magnetic fields theoretically, but solar eclipses allow them to be observed directly. Observations from the 2017 eclipse largely confirmed current solar models. Zurbuchen then discussed using eclipses to explore the outer solar system. NASA's New Horizons mission sent a spacecraft to observe Pluto, the Kuiper Belt, and even more distant bodies. To pinpoint Pluto's location necessary to orient New Horizons on its path to Pluto, NASA used a modified Boeing 747, which Zurbuchen described as an airplane that flies around a telescope that stays steady during flight. In 2017, the 747 flew across the Pacific Ocean and observed Pluto's occultation, its shadow cast by a star, across the surface of the Earth. Using data from that observation, NASA oriented New Horizons to successfully fly by Pluto in 2014. To locate a new target for New Horizons observation, scientists hunted for the shadow of Kuiper Belt object 2014 MU, estimated to be only 20 to 40 kilometers in diameter. First, NASA located a star which could cast 2014 MU's shadow across the Earth. After calculating the anticipated shadow path, NASA used more than 40 telescopes in separate locations to observe the star momentarily blink, indicating 2014 MU's alignment between the star and the Earth. With that data, New Horizons is scheduled to observe 2014 MU on January 1st, 2019. Finally, Zurbuchen discussed eclipses in other solar systems around other stars. Because planetary transits of other suns cause measurable dips in the brightness, scientists can identify the number and size of planets in other solar systems. That information also allows scientists to deduce a planet's distance from its star. In 2017, using this method, scientists identified 7 planetary objects in the TRAPPIST-1 system, 3 of which are in a location where liquid water can pool on the surface. Zurbuchen said that the TRAPPIST-1 findings are one of the most significant results from any NASA mission, not only because of what has been learned about the TRAPPIST-1 system thus far, but also because, as he said, the promise of the story for more, that there may be life elsewhere in the universe. Ultimately, Zurbuchen said we need to use spectroscopic methods to observe far distant worlds for signs of life. He said the James Webb Space Telescope will be able to make those observations, but even that will not be enough. To have the best possible observations, it will be necessary to create artificial eclipses using a star shade or coronagraph. Scientists and engineers are developing this technology now. After Zurbuchen concluded his talk, President Milstein invited questions from the audience. A member asked whether solar flares indicated by the TRAPPIST-1 observations are intense enough to eliminate the possibility of life on its planets. Zurbuchen said yes, solar radiation would wipe out any life on those planets. A guest asked about solar cooling on Earth. Zurbuchen said observations indicate both ultraviolet and visible light have recently been dropping in our sun. Historical evidence suggests this is not a unique occurrence in human history. NASA is currently creating a data series using observations from an instrument mounted on the International Space Station to develop the model for this current cooling period. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:41 p.m., President Milstein adjourned the 2,389th meeting of the Society to order at the social hour. Temperature, 2 degrees C. Weather, clear. Attendance, 106. Respectfully submitted, James Heelan, Recording Secretary.
Thank you, James. Are there any comments, corrections, addendums to the minutes? I have one. Maybe it wasn't at this meeting, but I believe in discussing the effects of solar flares on the TRAPPIST system planets, somebody suggested that the problem was only if life was at the surface.
That was at a previous meeting.
A previous meeting, okay.
Yes.
Then I stand corrected. So then, do I have a motion to accept the minutes as read? A second? All in favor? All opposed? Minutes are accepted as read and will be posted to the website in due course. We will now turn to tonight's lecture on Earth's earliest life. And it's my pleasure to introduce tonight's speaker, Andrew Knoll. Andy is Fisher Professor of Natural History at Harvard University. Previously, he was Associate Dean of Arts and Sciences at Harvard, and he also was chair of the Department of Organismic and Evolutionary Biology there. Currently, he also is a member of the science team for the Mars Mir mission. Andy's research focuses on the early evolution of life, Earth's environmental history, and especially the interconnections between the two, with a particular interest in understanding the history of oxygen in the atmosphere and oceans. Andy's honors include the Walcott and Thomson Medals of the National Academy of Sciences, the Oparin Medal of the International Society for the Study of the Origins of Life, the Phi Beta Kappa Book Award in Science, the Moore Medal of the Society for Sedimentary Geology, the Paleontological Society Medal, and the Wollaston Medal of the Geological Society of London. And he is a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the American Philosophical Society, a foreign member of the Royal Society of London, and he has an honorary fellowship in the European Union of Geosciences. He earned a BA in Geology from Lehigh University and a PhD in Geology from Harvard in 1977. Please hold questions for the question and answer period at the end of the lecture, and join me in welcoming Andy to the podium. Andy.
We're close.
We're close.
Just see what happens. Yes, good.
Yay. It's all
Okay.
yours.
Well, thank you, Larry. Thank you, everyone, for coming out this evening. When Larry asked me to speak, he said that they were interested in the origin of life, the history of life on Earth, life on other planets throughout the universe. And I thought, well, that may take more than 60 minutes. So what I'm going to do is, frankly, what all professors do. I'm going to talk about what I know, which is not much, but it's really about how we can reconstruct the history of life on our own planet. And again, just to give you a spoiler alert, is we live today not in a representative moment in our planet's history, but in really an end member of our planet's history. For most of our planet's history, life has been different than it is today. Environments have been different than it is today. And so we can only know about those, in some senses, alien states of the Earth by putting on our shoes as geologists, going out and interrogating rock records that preserve a history of that life. Then we'll take a little bit of time and say, well, if we can reconstruct the history of our own planet from the record of sedimentary rocks, then in principle we can go to another planet and look at sedimentary rocks there and reconstruct the history of another planet. And as many of you know, we've begun to do that on Mars, and I'll talk a little bit about that. And then at the very end, simply for purposes of provocation, I will try a slide or two on, are there things about life on Earth that we might consider general? What aspects of life on Earth do we think might be different from life elsewhere? And then we can have at it. So, I think all of you know that one of the great things about our planet is that it records its own history. And it records it largely in sedimentary rocks laid down one after another through time. And within those sedimentary rocks are patterns that tell us about ancient processes, both physical and biological, ancient states of the Earth system, and the nature of life at the time that those rocks were deposited. And as most of you are aware, we have a conventional history of life that paleontologists have given us over the last 200 years that is based on bones, shells, tracks, trails, and goes about 580 million years into the past. And as you can see here, whoops, in more recent times, we find very familiar animals on land, mammals. You go back somewhat further than that, life starts, it's still recognizable, but it's certainly distinct from anything we know today, the age of the dinosaurs. We go back to about 370 million years, and we actually see fossils that chronicle the ascent of life to land from its aquatic ancestry. And you go back to the beginning of the Cambrian period, about 541 million years ago, and we see the unfurling of the major body plans of animal organization that we see today in the oceans. Now, if we take that record and put it on a complete history of our planet, what we see is that the familiar fossil record of animals and plants is really only the last 15% of our planet's history. And that, of course, raises an interesting question, which is what could have come before? And comparative biology actually gives us some clues to what that might be. What you're looking at here on the left-hand side of the picture is what's sometimes called a universal phylogeny, or a universal tree of life. And it is a hypothesis of the genealogical relationships of all organisms, from bacteria to us, really based on the comparisons of DNA or protein sequences among species. And there's certain aspects of this tree that are not controversial. For example, it has 3 major branches. Over here is the bacteria, which are pretty much as advertised. Here is our own branch, the eukaryotes, that is, organisms with cells like our own, in which the genetic material is bounded within a nucleus. And then there's another major branch of the tree called archaea, which, like bacteria, has a very simple organization, but actually differs from bacteria in a number of important ways, everything from the nature of the lipids and membranes in these cells to the way they actually translate and transcribe DNA. The archaea were only recognized as being separate in 1977, and we're still learning a great deal about their evolution. And one of the interesting things is you can see that early on we seem to have a split between bacteria and archaea, and then our own group, the eukaryotes, arises at least in part out of the archaea. It probably actually arises out of a partnership between archaea and bacteria, but gives us a separate evolutionary trajectory. The other important thing to note is if we ask, where do we find in this inferential history of life that we get from comparative biology, where do we find the record of the animals that make up the conventional evolutionary history? And the answer is they are simply on the shallowest branches of one of the shallow limbs of the tree of life. The inference then is that there was probably life on this planet well before the origin and diversification of animals, and that that life was very likely microbial. So if that's the inference, then the next question is a very practical one. Is it possible that bacteria, so small and fragile and simple, can actually leave a tractable record in rocks that we can actually trace back through time? So to test that hypothesis, we're going to actually look at some rocks from a place that I worked on a lot when I was younger and fitter, and that is the record of Spitsbergen, which is about halfway between the northern tip of Norway and the North Pole. And you can see for geologists the reason to want to work there is that sedimentary rocks are extremely well preserved, and the rocks that you're looking at here were deposited between about 730 and 800 million years ago. Each of these color swatches is about 300 meters or 1,000 feet thick. So it's an extraordinarily good library of Earth history at a time which is more than 100 million years before we see even the first glimmers of animal biology. So the question then is, if we look at these rocks, do we find any signatures of life that we can actually interpret? Well, this being the Philosophical Society of Washington, you've all probably figured out that I wouldn't have brought it up if the answer had been no. So what we find is if we go and look at some of these rocks more closely, these are limestones, calcium carbonate, and you can see the bedding here is just this kind of wavy lamination. Now, the geologic record is a record of pattern, and because we can observe pattern formation on the present-day Earth, and because we can do experiments, we can actually look back at that pattern and interpret it in terms of state and process. In this case, if you want to see sort of wavy laminated carbonates like this, you go to a place like the Bahamas, and that wavy lamination almost invariably reflects microbial communities forming a mat on the surface of shallow water, sort of coastal environments, and then that interacts with sediments, sands, and muds as they move through the system. That's exactly what we think is going on here. If you look closely, you'll see that in the middle of this picture, there's a big hump. It looks like a teepee, and in the button-down parlance of geology, that's actually called a teepee structure. And it is, in fact, not like a teepee, it's actually a ridge, but these ridges form in areas at the sort of the driest end of a tidal flat where sediments are left high and dry, pore waters in the sediments evaporate and precipitate calcium carbonate minerals, and then that pressure actually causes the sediment to buckle. So the whole point is that with a little bit of training, you can actually read something about the environment at the time these rocks were deposited and get some preliminary inferences about what kind of organisms might have been there. Now, we really are helped by these black structures here, which are concretions. They're made of silica, SiO2. It's the stuff of quartz, and this is usually called chert or flint. And what happened is that soon after these rocks, or these sediments were deposited, these nodules actually grew within the sediment, and they incorporated materials that had been deposited earlier. And they actually preserve things on a micron scale. So if we take a piece of this rock, cut a paper-thin slice of it, and look at it under the microscope, what we see is something like this. These are cells. Each of these cells is about 4 microns in diameter. These are certainly bacteria, probably a particularly important group of bacteria called cyanobacteria, which I'll talk a little bit more about in a bit. But the important point here is that indeed, even tiny evanescent bacterial cells can get preserved under the right circumstances and give us a record of life that potentially will extend far back before the evolution of animals. And in fact, when you look at different types of rocks from different environments in this succession in Spitsbergen, you find a lot of stuff, the equator on Mars, a place called Meridiani Planum. And you might ask yourself, how does NASA actually decide where to put this little rover on the large area of Mars? And there's at least 3 criteria. The first one, as I already said, this rover is solar powered, so it had to land within 15 degrees of the equator. The rest of the planet's off limits because it couldn't keep its power supply up. The next issue is safety. And so if you asked a geologist, where would you go in North America to learn something about the geological history of the planet? You know, she might say, oh, the Grand Canyon, because they have all these rocks exposed. If you ask an engineer, where would you like to land? The engineer will say Kansas. And let me tell you, when the machine costs $2 billion, the engineer always wins. Fortunately, Meridiani Planum is both scientifically interesting and feasible from an engineering point of view. This is Meridiani Planum. What you're looking at here is the calculated uncertainty ellipse for landing. It's about 75 kilometers long, 10 kilometers wide. That basically is calculated from Earth, and we did in fact land right about there, which is, I once actually worked this out, it's equivalent to hitting a golf ball from Boston and hitting a hole-in-one in San Francisco. So it's pretty good engineering. Now, what you can see here is that this is flat. Meridiani Plana was one of the flattest surfaces in the solar system. So engineers are happy. Geologists are happy because the colors that you see here is a heat map for the abundance of the iron oxide mineral hematite, Fe2+3. It turns out it's very difficult to know how to spread hematite over the surface of this plane without using water. So that was the beacon that brought our rover to this place. Now, the rover landed on January 24th, 2004. It unpacked itself. It looks around and takes some pictures. And I must say, one of the most exciting moments in my scientific life was when we saw the first pictures of where the rover landed. It landed in the middle of a small crater. This is the crater wall. And even from these first pictures, you can see there are layered rocks. Now, to a geologist, that says there's history. And we can maybe understand that history. When we got up close to it, we saw that these are not only thickly bedded, but they are laminated on a millimeter scale. These are sedimentary rocks. When we look at them with the hand lens, they are sandstones. They're full of sand, sand grain particles, and they are in single grain layers of a type usually associated with deposition by wind. So most of these sands were deposited by wind. On the other hand, every once in a while, you'd see a departure from this basic lamination. So if you look up here, you'll see there's these scallop structures on a millimeter scale. Those are what are called ripple marks. Here is some in cross-section in the modern world. And you've all seen ripples forming along a beach or the side of a river. And though the particular kind of ripples that we see here are very diagnostically associated with deposition in a liquid. And water is the only game in town. So this tells us that there was water flowing, at least transiently and episodically at this time. We can then put our scientific instruments to work on these. And the first thing is what we didn't see. So if we look at the Mossbauer spectrometer, which looks at iron minerals, the important thing to know is that the entire surface of Mars, to a first approximation, is just covered with basaltic volcanics. That is the kind of volcanic rocks you find in Hawaii, for example, today. But interestingly, when you look at these sediments with the Mossbauer spectrometer, you don't find the iron-bearing minerals that are generally associated with basalts. There's no olivine, there's no magnetite, there's no other minerals that carry iron. In fact, the main iron-bearing minerals are something called jarosite, which is a sulfate salt, hematite, iron oxides, this thing called FeD3, which is just a funny technical term, but we know from modern studies that, again, that's generally associated with particular types of iron sulfates. And in general, when we look at the elemental composition of these sediments, you know, most of the ions, the cations, are the things you would expect to find if you had actually dissolved basalt in water. Calcium, magnesium, iron, aluminum. Most of the anions, as the negatively charged ions, are actually sulfate, which actually comes from volcanic gases. And interestingly, basically, these rocks regionally have the elemental composition of basalt plus sulfur gases. And there's nothing in the history of these that actually fractionated elements. That is, there wasn't enough movement that some of the iron went out here, some of the calcium went out there, some of the magnesium went out somewhere else. So that tells you that this is a local environment that probably doesn't have a whole lot of water associated with it. The other important thing is this mineral jarosite, which is, I think, quite a wonderful mineral. The reason is that if you want to find jarosite on this planet today, you would go to an acid mine drainage, because jarosite only forms in strongly acidic environments. So that tells you something about the nature of early Mars. And then finally, there are some, I talked about concretions, things that grew in the sediment on Earth, those chert concretions. Well, we have concretions in these sedimentary rocks on Mars too. Here is just some cobbles of the sedimentary rock. You see the lamination here, and you can see that sticking out from the rock are these things that we call blueberries. Now, I should say that they look blue because this is a false color picture. If you use true color when you're working on Mars, you find that the entire Martian surface is colored by subtle variations on orange. You just don't see much. But by looking at different wavelengths and putting them together in different ways, we can actually get false colors that really emphasize the difference between chemically different things. So these blueberries, a couple of millimeters around, here you see them with our hand lens. They're little nodules. And in fact, when you look at just the surface sediments around these, they're covered with blueberries. So we wanted to know what these things were made of. Now that's kind of an interesting technical issue, because the size of a blueberry is much smaller than the spot size of our chemical instruments. So we finally found this place called the Berry Bowl, where we could look at this area, which is blueberry free essentially, and this area, which was full of blueberries, and by differencing them, get some sense of what these blueberries were made of. And the punchline is that the blueberries are made of hematite, Fe2+3. And at that point, it became abundantly clear that this signature of iron oxides that we saw from orbit is basically these blueberries being weathered out of sedimentary rocks and just being distributed regionally on the planetary surface. So we know something about sediments, about the physical nature of these sediments. We know something about their chemical nature. And as we started traversing around, we found that in larger craters, we had larger exposures of rock. On Mars, meteor craters play the same role for geologists that basically road cuts and cliff faces play on Earth. So here's a larger crater called Endurance, and now you see there's a lot more exposed. You can see this laminations, very much like we saw in our initial crater. If you look at the bottom here, you'll see that there's laminations, but they're running at an angle by some 30 degrees or more. And it turns out that if you follow these up to the top of this exposure, you will see the kind of ripple marks that tell us that there's water present that we saw before. Now, again, the way we interpret these kinds of patterns depends on looking at places, in this case on Earth, that have similar patterns where we can relate them with confidence to processes. So here's a place in Namibia, southwestern Africa, a place called Sossusvlei. It was a marvelous place with 200-meter high sand dunes that migrate along the coast. And my friend and longtime field partner, John Grotzinger, and I were walking through this little ravine, and here you see the same motif that we saw on Mars. Here are these beds at an angle called cross bedding, a couple meters thick. Above them, there are similar laminations, but now flat-lying, which are usually called aeolian sheet sands. And at the very top, there is sediments that are influenced by water. Precisely the same motif that we saw on Mars. These are the signature of migrating sand dunes. These are also aeolian, and then there are transient lakes that gave rise to some, at least transient liquid water. So again, we can actually say something about environments in detail. And this, I'm very proud of this paper that John led, because this was the first stratigraphic section ever measured on another planet. It's only about 7 meters thick, but it's Mars, for God's sake. And here you can see at the bottom is the aeolian dune field, aeolian sheet sands. We can see evidence of groundwater percolating upward through these, and it actually made it all the way to the surface at the top. And here is just a nice picture looking from the bottom of this crater exposure. There are the wheels of opportunity. Here is where Oppie took chemical samples on the way down. So again, we can do this, virtually do much of the same kind of exploration on Mars that we saw on Earth. Okay, so the history then, we don't really know the age of these rocks except that they are old. Estimates of age on Mars are based on cratering statistics, in turn based on the moon. And the estimated age for these sedimentary rocks is on the order of 3 to 3.5 billion years old. There's evidence for water regionally because of these chemical interactions that we saw, laterally because some of the features that we see. There's evidence for groundwater percolating. You couldn't get those blueberries unless you had groundwater percolating through the sediments. So we know there's water there. On the other hand, we know it's arid, and because of that jarosite, we know it's oxidizing. So 3.5 billion years ago to 3 billion years ago, this place on another planet we know is arid, oxidizing, and acidic. So let's ask the big question then, which of course the news of the world was way ahead of the conventional press here. We know something about the environments of Mars at this time. Can we say anything about life? And I should make it clear that no one has ever found any signature of life on Mars. That doesn't mean it wasn't there, but nobody has found anything compelling yet. On the other hand, we know something about it, and we can, by looking at environmental analogy on Earth, ask about the likelihood of life. That is, let's go to places on Earth where jarosite is common. Do they support biology? And then, NASA's mantra for a long time has been follow the water. But you can't just follow the water. You have to ask about its properties, because not all water is the same. And you have to ask about its persistence. If water is present for 1,000 years and then gone for the next 10 million, that's not very interesting biologically. If it's the reverse, it's very interesting. So let's very quickly, there's a place called Rio Tinto in Spain, which I've worked at off and on for the last 15 years or so. It's a wonderful place because it has a Tinto Rio. The water is the color of rubies. It's the color of rubies because the pH varies between 1 and 2. And at that acidic conditions, you have ions of iron sulfate that are actually red colored and are stable in seawater, or in the water. And you can look at this orange stuff here. It does precipitate minerals. We've actually looked at them with the same Mossbauer spectrometer that's been used on Mars, and I will tell you that the mineral composition of these is almost identical to what we see at Meridiani Planum. A very similar system chemically. Well, is there life there? Yeah, it's full of microorganisms. No problemo as far as microbes are concerned. And not only that, but they actually fossilize in iron oxides. So if there would be life in a place like Meridiani Planum, it may well have left a record at a size scale that wasn't available to us. So that doesn't help us much. But there's another feature that I think may constrain our thinking about life. It's something called water activity. Now, water activity scientifically really comes from the idea that when water is present and there's a solution, there are solutes in it, that the water actually forms weak transient bonds with the ions in solution or organic molecules in solution. And when those water molecules are bonded, they're not able to do the other chemical work of water. And so in a sense, water activity is a statistical sense of how much of the water in the solution is available to do the work of water. For our purposes, you can kind of think of it as salinity. And I talk about it in terms of the ancient mariner's plight, because I think Samuel Taylor Coleridge really helped us to understand what I'm trying to get at here. You've all were forced to read The Rime of the Ancient Mariner in junior high school, right? And you learn that the mariner is in his dinghy and he is surrounded by water. There's no land for hundreds of kilometers in any direction, and yet he's dying of thirst. And why is a mariner dying of thirst in the middle of water? Now, Coleridge, of course, tells you it's because he shot an albatross, but scientists know it's because the water activity of seawater is too low to sustain for long periods of time the life of a human being. Okay? Now, there's been a lot of work on water activity, and you'll soon see why. Pure water would have a water activity of 1. Seawater is about 0.98, and most of the life you've ever seen or experienced in your life goes on at water activities between about 0 and 0.95. Well, let's go down a little bit. Now we're getting to more concentrated solutions. Here's orange juice concentrate. Now you might ask yourself, who on earth was it who bothered to measure the water activity of orange juice concentrate? But it turns out that the entire science of food preservation is based on water activity. Now, your ancestors figured out thousands of years ago that if you salt meat, it doesn't decay. And people in Provence cook fruit in sugar syrup, so it gets a water activity that simply won't support microbial life. So here's orange juice concentrate. Here's a saturated salt solution, and there are a few archaea and fungi that can metabolize at that. And then you get to the very bottom. The world champion, so far as we know right now, is a little fungus called Xeromyces bisporus, which can actually carry out its life cycle at a water activity of 0.61, which is really hellacious. Now, you've all done a astrobiological experiment in your own kitchens, probably without realizing it. You open a jar of honey, you take some out, you put the jar back on the shelf, and then you forget about it for 10 years. And when you get back, will the honey have gone bad? The answer is no. If you put water in it, it'll go bad in a few days. The reason it doesn't go bad is that honey has a water activity that's lower than the limit to life on this planet. So if you can understand water activity, that can put some very sharp constraints on our thinking about biology. So let's do that for just a minute or two. We're going to start out with a simple experiment. You're going to take a beaker of water, put some salt in it. It'll dissolve. And then we're going to let that water evaporate. So as the water evaporates, the concentration of the sodium and chloride ions go up. They go up to a certain point, and then they stop going up because you start precipitating table salt out as a mineral, which shows that when you have high concentrations of ionic solutions, very few organisms that we know of are able to tolerate this. So what this tells us is that Meridiani Planum, yes, there was water there. There had to be water there to give us some of the physical and chemical observations that we make. But that water then started to evaporate. It basically evaporated to a point where no life that we know anything about could tolerate, and it never came back. So the simple fact that Meridiani had water does not tell us that it's a bio-friendly environment. And just one other thing, there's another way to get at this, which some nice work done by a guy named Nick Tosca, now at Oxford, who was a very good postdoc with me. The Spirit rover, it essentially developed a game leg at some point, and it was just pulling its one wheel through the sediment and made a wonderful discovery because it kept dredging up this white stuff. And this white stuff, which is probably somewhere between 3.5 and 4 billion years old, is amorphous silica. That is, it's SiO2, the same stuff as quartz, but it's not crystalline. And everybody says, oh, that's great. And there's a paper on how you need water to make amorphous silica, and that's true. On the other hand, if you know anything about silica on Earth, is that there's hardly any amorphous silica on Earth that's more than 1 million years old, because if it keeps interacting with water, it crystallizes as quartz. And what I say Nick and I, but Nick did all the work, what he really found is that there's lots of minerals in these very ancient rocks on Mars that are simply not stable when they are in the long-term presence of water. So again, that tells us that Mars was at least at some times in some places wet early in its history, but it's been dry for a very long time. Geomorphology would tell you the same thing. This, for example, from the work of Bethany Ellman at Caltech, is an old delta system. So there were lakes and deltas, but they're all old. And once you get to 3 billion years old, Mars seems to be pretty much as dry a planet as it is now. So the takeaway message for Mars then is, on the same time scale that life was taking root on Earth, the window for life was closing on Mars. So our neighbor has a very different history from ours. Second to last slide then. I feel I have to put this in just to thank the people who made the mission possible. You're looking at the sun setting over the Martian horizon. And mission success for this mission was defined as 90 Martian days of continuous operation plus 600 meters of wheel traverse for the two rovers combined. Spirit gave up the ghost in 2010, having gone 2,210 Martian days and going nearly 8 kilometers. Opportunity is still going. It was today was its last planning. It's gone more than 5,000 Martian days and has more than completed a marathon. It's gone 45 kilometers. And that's really wonderful engineering. So I promised you a final slide just to get you all riled up so you can ask me questions. The only life that we know about in the universe is us. So if we want to think about generalizations, the kind of thoughts that you might think about at a Philosophical Society of Washington, we have to ask ourselves, is there any way that we can think about generalities based on an example of one? And I would argue that there are a few. I think if and when we find evidence of life on the planet Remulon, it will be based on carbon. Carbon has an unusual chemistry that allows it to form very versatile bonds that make possible literally millions of different kinds of molecules. The only other element that you might think would be similar would be the one that's in the same column of the periodic table, which is silicon, but almost all silicon in the universe is tightly bound to oxygen, and it just doesn't participate in the same kinds of chemistry. NASA's measures follow the water. Is water the only solvent for life? There's been some essays on this, and the answer is, in a technical sense, no. Water has some very interesting sort of biofriendly characters, and there are a few organic molecules or even ammonia that under the right circumstances might preserve a record, might actually permit biological reactions. But for most planets, rocky planets, probably if there's life, there's water. And then, well, it disappeared, but oh, there it is. The only other thing to mention then is I would think if life persists for a long period of time, you have to have complementary metabolisms. That is, if something takes CO2 out of the atmosphere or water environment, something else must put it back in. So we need that. And then finally, there's the challenges. You know, it's very nice to talk about these, but if you actually want to explore astrobiologically, you can sort of divide the universe up into 3 parts. There are our own solar system, where, as we've done on Mars and can do in principle in other places, we can go there at least virtually. In nearby solar systems, where we now know most of the stars in nearby solar systems have planets, we might be able to learn something about life by learning about the chemistry of their atmospheres, that there may or may not be a diagnostically biological atmospheric signal, but that's pretty much what we can look at. And really, for the rest of the universe, the only way we're going to know if there's life in the rest of the universe is if they call us. So, and with that, let me thank you for your attention and try to answer questions. So, questions?
Well,
Yes.
thank you, Andy. We have we have time for some questions. A little ahead of myself here. We have a process for the questions so that people listening on live stream can hear it, and it'll show up on the recording. There are some runners with microphones. The microphones are purple and red and green. And if you'll raise your hand long enough for somebody with a microphone to see you, they'll bring you the microphone eventually. And when you get it and you're called on, please stand up, tell us your name, tell us if you're a member, and ask a question. Save the dialogue for afterwards.
Yes. Well, there's...
Red microphone.
Oh, yeah. Hello, everybody.
Name?
My name is Chris Sorensen. I sadly am not a member, but I definitely hope to be one someday. First of all, thank you for your awesome talk. It was really exciting and informative. It's always exciting to hear about Mars. My question is, what do you think about the prospects of finding life on other worlds, like the ice worlds, like Enceladus and Io, and how might your work apply to that?
Yeah, that's a really good question. Mars is not the only part of our solar system that is interesting biologically. It is probably the planet where, at least in the lifetime of my grandchildren, we're going to be able to do the kind of virtual field work that we do here. But on moons like Enceladus, where you have, what do you call them, Old Faithful, geysers, right. There is a plan, particularly Carolyn Porco is leading a group that wants to basically fly a low orbiter in and actually sample the materials coming out of the geyser. And, you know, if there's life there, you may well find diagnostically biological organic molecules. Again, in all of this, there's always a worry about false positives, so one has to be, you know, really very careful in this. But yeah, I think certainly in Enceladus, if we could figure out a way to actually drill through the ice over the oceans in Titan, it's worth looking there. I don't know that any of these have a very high probability, but the probability isn't zero, and they're certainly worth exploring.
Next question. Okay, green microphone.
Sylvia Valentine. And I was curious, how similar do the ancient anaerobic types of organisms, are they similar or dissimilar to the ones that we find in plate tectonics?
Okay, so there's kind of two questions bound up in there, and I guess let me interpret it, and if I get it wrong, clarify it. And that is, you know, today we see plate tectonics, we see different types of organisms, including those that live without oxygen. And if we go back to 3 billion years ago, are those sure guides? Is that a fair way? And the answer is maybe. I think there are things we can say. We actually have a group at Harvard that meets every week to talk about the origins of plate tectonics. And I think it's pretty clear that the physical engine of the Earth has worked, at least in a broad way, like it does today, for more than 3 billion years. And certainly, I think that anaerobic microorganisms that were living on the oxygen-free Earth had a biochemistry that is broadly similar to those that we have today. Whether, you know, there was actually methanococcus or something like that, we don't know. But I think at the broad level of metabolism, I feel fairly confident that we can extrapolate that backward.
Let me just make one comment. If you're watching the live stream, you can please ask your questions using the comments box. And you're monitoring them, Savannah? Okay. Carl, with the green microphone.
Hi, Carl Merrill. I'm a member of the Society. I really enjoyed your talk, and I thought it was a nice presentation of life as we know it on the Earth. But given that this is the Philosophical Society, I'm forced to ask a question.
It's PSW science, Carl, and you know that.
Ludwig Boltzmann was one of the first to realize that things were made up of atoms, and I understand that he realized that he was made up of atoms, and then atoms can think. So we know that there may be a chance that silicon and things we constructed can think. And so then the question is how you define life. And if we're just an intermediate phase, and then you get to another phase, maybe that other type of life is even more common if you define that as life. So
Is there a question? What was the
Yeah, I think I'm going to interpret it as a question. In a sense, this brings us into the future, that do we get to the point that, Steven Spielberg got to 20 years ago, where we actually have, in silicio, things that can think and react and have emotions and everything else. And there are probably people in the room who are better than I at knowing how realistic that is. I think the definition of life that I like the best is one that Jerry Joyce from the Salk Institute once articulated, which is that life is a chemical system capable of Darwinian evolution. And I like that because it basically says that evolution isn't some sideshow to biology. Evolution is biology. Evolution is the major thing that we need to understand. And so I guess if we can get an in silicio system to the point where it actually can evolve, then we should probably worry. Whether or not we would get there, I don't know.
I should tell everybody that Jerry Joyce was a speaker not too long ago in the fall, I think in December, and his video has been posted. So if you're interested, you can see Jerry Joyce's talk on our YouTube channel. We have one in the back there. Is that with a purple microphone?
Yes, hello, I'm Marie Burton. I'm not currently a member. This follows on Dr. Carl's question. He's the expert on the subject, but we've talked a lot about the biodome. Have you considered viruses?
Yeah, viruses are absolutely fascinating, and we were talking about these at dinner tonight, that what we have learned about them in the last 10 years is truly remarkable, and it really suggests that we really have an immense amount to learn. There are people who are biological oceanographers who think that 90% of all the genes present in the ocean are present in viruses and not in cells. So the question then is, what is the relationship of viruses to cells? And by definition, a virus doesn't exist without a host to actually replicate its DNA in that. So in some ways, if you take Jerry Joyce's definition of life, viruses do evolve. That's why we need new flu vaccines every year. And I'm happy with this definition to think that viruses are alive. On the other hand, they're not metabolizing, at least most of them aren't. There are things that cellular life does that viruses don't do. So I tend to think of viruses as a rogue form of life that's completely dependent on cellular life. I don't know what you think of that, Carl.
No, if you're going to talk, use the microphone, and we try not to have a dialogue here.
I think that I look at it as a whole system.
Oh.
So the viruses aren't independent
Right.
from the cells. It's all one big system,
Right.
and it all evolved together.
No, I think that's consistent with what I said. There's cellular life, and then there's this stuff that takes advantage of it. And I'm happy to consider that living. So, anyway.
Tim, with the green microphone.
Timothy Thomas, I'm a member. I'm wondering about when the signature of life in the evolution of the Earth was completely obvious, as it is now. As we're looking at exoplanets, how old does the exoplanet have to be before it's as living as Earth is right now?
So you're saying at what point in the history of the Earth was it obvious that life was there?
Correct, especially if we're looking at it as an exoplanet in our atmosphere and some chemical detection on the surface. Perhaps.
Okay, let me give you a couple of approaches to an answer and see if any of them are actually addressing what you're trying to get at. There's no question that the only signature of life we are likely to get from an exoplanet in the next 50 years is a signature of its atmospheric composition. And there is no question that if you define a biologically influenced atmosphere as one that contains oxygen and methane at the same time, that's a favorite NASA idea, that you would have a false negative for the first 2 billion years of Earth history. So I think there's a very real question as to whether there is any atmospheric signature that can only be made by biological processes. I once had dinner with a guy named David Stevenson, who's a very prominent planetary scientist at Caltech. And I sort of gave him this line about, well, if you have oxygen and methane, since they can't exist in equilibrium, if they're both there, it must be biological. He just kind of laughed. He says, you know, I can guarantee you that within a year of some probe making that measurement, there will be 10 models of how to do it without invoking biology. And I suspect he's right. And I think most of my friends who do extrasolar planets are not highly optimistic that there will be a biological signature. There's certainly ways of making oxygen that don't involve life, particularly on the right kind of planet. I'm not sure, is there more to the question that I should?
Well, the question is about if, in other words, you're saying even today, if you looked at Earth, you could not be convinced there was life on it from a sufficient distance.
Well, that's
It's a pretty living planet.
That's an interesting question. Of course, we're burdened with the preceding knowledge that there is life on Earth. So we tend to actually look at it from afar, and we actually have images of Earth by Viking and Mariner and things like that. We see things that we know are signatures of life, the oxygen on the planet, because we know on this planet the oxygen comes from photosynthesis. But I think if we didn't have that prior knowledge, it isn't clear to me that that would be an unambiguous signature. I guess the other part of the question is, if you ask as a geologist, at what point do you have evidence of life, what we might see on another planet? There it becomes kind of interesting, because I keep telling all my students that the Earth is actually writing its history in sedimentary rocks, but it's writing with its left hand and it's erasing it with its right. And so when you get back to the early Earth, we really don't have a very good record. And so I think much of our ignorance about early life on this planet comes from the fact that the record is not that good. And that's where Mars becomes interesting, because Mars doesn't have an active plate tectonic interior. So there are rocks that are 4 billion years old sitting at the surface of Mars, and they are as well preserved as 2 million-year-old rocks are on the Earth. So in some ways, if you're looking for a geologic history of life and or environments, sort of tectonically quiet planets like Mars may be better than Earth.
So I want to interject a question.
Sure.
So Lee Cronin has been proposing that you can determine whether there are living things on wherever on the basis of the presence of chemicals of a certain level of complexity. It's not a particularly new, not necessarily new, and Jim Garvin gave a talk last fall in which he had a matrix of various parameters, but Lee has an index of chemical complexity, and he says you can't find a molecule that has a higher number on the index than a certain threshold number. And so if you detected any chemical structure with that level of complexity, it will be a sure sign that there's life.
Yeah, again, as always, the answer is maybe. I think there's a number of things that have to be unpacked. One is you can go to, there's a class of meteorites called carbonaceous chondrites, which have up to 5% organic matter in them, and there are lots of complex organic molecules.
They're not that complex.
Well,
he says they're not that complex.
Well, there's a couple of issues there, some of which have to do with what gets analyzed and what gets reported. So we know that physical processes are capable of making complex organics. Even in the interstellar medium, it's pretty clear that we're limited on the complexity of the organic molecules we see by our ways of detecting them, and we don't necessarily know what the limit is. Now, that said, if you show me an amino acid from the planet Remulon, I will not get excited, because I know that any high school kid can make amino acids in an experiment that you can do in school. Again, if you show me cholesterol, I'm
That's his
interested.
point.
So
That's his
I think there certainly are molecules that, to the best of our knowledge, are associated pretty resolutely with biology. We might be able to see them in our own solar system for planets in solar systems around other stars. I think that is, at the very best, very far in the distance.
So we had some other questions here. I guess Bruce with the purple microphone. Yep, that would be you.
From what we gathered from your lecture.
Name, membership?
Can you hear it?
Yes.
Yeah.
Your name.
So did RNA or something very similar to it thereby exist really long time ago? And how might it ever have arisen at such a complex bunch of chemicals? Does that?
That's a very good
suggest
question,
this?
right? And I guess when Jerry Joyce was here, he probably talked about some of the lines of evidence that are associated with this. The reason people like RNA is because there's this very deep chicken and egg problem of life on Earth, and that is you need DNA and RNA, you need nucleic acids to make proteins, and you need proteins to make nucleic acids. And so if you had one molecule that could actually be an informational molecule like DNA and also have catalytic activity like a protein, that would be pretty interesting. And RNA has those characters. That's why Tom Cech won the Nobel Prize. And in fact, a little bit later, many of you know that the place where RNA, sort of messenger RNA, is made into proteins is a ribosome. And Tom Stites down at Yale got the Nobel Prize for showing that even though there's a lot of proteins in a ribosome, the functional heart of a ribosome is RNA. It's a ribozyme. So I think there is a I think, important school of thought that says the earliest self-replicating evolving systems on this planet may well have been RNA or something like that. And we know that RNA can evolve in the laboratory to take on new functions. It's a very interesting molecule. Now, how you make it has always been a problem, particularly because, you know, its constituents are sugars. You can make those. People have known how to make sugars from inorganic processes or precursors since the 1860s. We know you can get phosphate in solution. We know that you can make the different bases that are involved in nucleotides. But until recently, nobody could figure out how to stick those things together to make the nucleotide, the basic building block of nucleic acid. And then in 2009, a guy named John Sutherland, now at Cambridge in England, figured out a very interesting pathway where he could make nucleotides. And we know that we can condense them together to make at least fairly short informational macromolecules. And I think what's really happening now is that the people who do these experiments are really starting to take on board the idea that all of this happened in the context of a planet in its early history. So people like John Sutherland are now putting forward hypotheses on how things like meteor impact could drive chemistry. And with a combination of theory and experiment, I think many of these things can be tested. So yes, I think one of the central ideas, not universally accepted, but a central idea in chemical evolution is that this sort of jack of all trades molecule RNA was an early part of the genesis of life.
Red microphone.
Hi, Nish Patel. I'm not a scientist, so...
Are you a member?
No, I'm not a member either. I'm definitely not a member.
By the way, his name was Bruce Murray, and he is a member.
Okay. So thank you for the 2 slides with the takeaways, because those made sense to me. But I had an observation. You talked about how the atmosphere of the Earth has evolved, and the changes in the atmosphere gave support to life as we know it, plants and mammals and whatnot. So my question is, as the atmosphere continues to change, will that support new life forms, and will the Earth be blessed with better life forms than human beings?
I think Isaac Asimov might have written more about this than I'm capable of doing. Yeah, I mean, the premise of your question, I think, is unassailable. That is, that as the Earth's atmosphere has changed in the past, it has, at least in the long term, changed in ways that have given rise to new opportunities for physiologically distinct and functionally distinct types of organisms. Whether that can continue, I don't think we know. But it's worth mentioning, because we're in Washington, that we also know from the geologic record that there are short intervals of time when the atmosphere has changed in ways that were not good for life. So 252 million years ago, in Siberia, there was volcanism that was about 1 million times larger than anything ever witnessed by a human being. And see if you've heard this story before. They put a lot of CO2 into the atmosphere. It caused global warming. It caused ocean acidification. It caused a decrease in the oxygen content of the deep ocean. And about 90% of all the species in the ocean disappeared. So in that sense, if I was going to make a prediction about atmospheric change in ways that will influence biology, I wouldn't make a prediction about 100 million years from now. I would make a prediction about 100 years from now, when I think we could lose a substantial portion of the diversity we have today.
Well, on that cheery note, I think we'll close. But before you go, I'd like to present you with a small gift and thank you for coming all the way from Boston and giving this wonderful talk. It's a framed copy of the announcement of your talk. And as a bonus, we also have a signed copy of the Bulletin of the Philosophical Society of Washington, Volume 1.
Great.
Which is really important because it explains the name.
Yep. Thank you. Appreciate it.
So before we go, there are just a few closing announcements.
Computer will still be there.
Just remember, if you're a member, please pay your dues. If you're not a member, you can join. Not that hard. You can just go to our website and go to the little tab that says membership, which will pull up the application for membership.
Yes.
Go down to the bottom after filling it out and answering a few simple questions. And press the submit form, which will bring up the donate to PSW form. If you donate, you can use PayPal, but most people don't want to use PayPal. You can just press this button and use whatever debit or credit card you like. You do not have to use PayPal. And pay your dues. And if we decide that we don't want to have you as a member, highly unlikely, we will refund. So it's a pretty easy process, and I urge you all to apply for membership. Remember that PSW is a nonprofit educational organization. It's tax exempt under Section 501c3 of the Internal Revenue Code. And contributions are tax deductible, or perhaps a little less so after passage of the new tax law. Our next lecture on the 2,392nd meeting of the Society will take place on April 6th, right here in the Powell Auditorium. The speaker will be Don Norman of UCSD. Don leads the design lab at UC San Diego, where he is also Emeritus Professor of Cognitive Science. He's well known for several seminal books on design theory, especially his book, The Design of Everyday Things. Don was, among other things, the Vice President of Apple Computer in the Steve Jobs days, and founded the Department of Cognitive Science at UCSD and served as its chairman. He will be speaking on democratizing design. Think the Internet of all things for everybody, designed to need. Oh, that's the wrong one. On April 20th, we'll be having Yuval Grossman, Professor of Physics at Cornell University. He'll be speaking on any matter, in particular, why there doesn't seem to be any of it anywhere. I should mention that Yuval is a good friend and colleague of two previous speakers, Nima Arkani Hamed and David Kaplan, and comes to us highly recommended by Nima. So if you like Nima's talk, you're very likely to like Yuval's talk as well, and I urge you to come and listen to him. On May 4th, we have Thomas Budavari, professor of applied mathematics and statistics at Johns Hopkins University. Tomas works on integrating very large, independently developed data sets, including large astronomical data sets. But lately, he's been applying the statistical and computational methods he developed for astronomy to the integration, analysis, and use of city data sets, such as water use, electrical consumption, road repair, demographic data, and the like, to develop more accurate and complete understanding of land use patterns to guide policy formation and implementation in our northern neighbor, Baltimore. Finally, on May 18th, we have the Cassini special, which we're still working on. And we were almost going to have Carolyn Porco, but that didn't quite work out. So we are still putting together our panel, and more on that later. The social hour ends at 10:30, after which PSW members and their guests typically go across the street to continue the discussion at the Fairfax Hotel Lounge, which I understand we're now in the lobby because the lounge is being renovated. This is because of our rowdy behavior over there, which caused much damage. It's just across Massachusetts Avenue, and if you want to join us, please speak with corresponding secretary Robin Taylor at the back of the room, or membership director Lloyd Mitchell, who's sitting up here in the front with the black vest on the white shirt. Please use the side entrance to exit the building, and I will now accept a motion from a member to adjourn the 2390th meeting of the Society to the Social Hour. Do I have a motion? We always have a motion from you. We have a motion. Do we have a second? You can't second your own motion. All those in favor? Aye. All those opposed? The meeting is adjourned to the social hour.